Hello, everyone, and welcome to the official kickoff the 2025 NEMIS Academy season. My name is Alina Bonadonna, and I'm delighted to be a webinar moderator of today's session. And of course, as always, we are starting in this season and in general our webinar with presenting amazing speakers who are joining us today. And today I'm super honored to present you Bruce Urtz, who is a director of microbiology at Sterilex. Hi, Bruce. How are you?
Hello, good.
Where are you based, Bruce, by the way?
So I'm located in Hunt Valley, Maryland. Um, been working with Sterilex for the past 3 years.
Cool. And how is the weather now there?
Cold,
Cold.
very cold.
Yeah, the same in Switzerland, I should say. Exactly the same. And just a very, very quick question, not to take too much time of presentation from Bruce. Do you guys hear us well, see us well? Just drop us reaction, the one you always see on the bottom left corner. I see that attendees are quite aware of our webinar rules. And while you are shooting on reaction, I just want to give my honest honest thanks to Nemis Technology for supporting this super young but quite powerful educational project in a food safety field. So, Bruce, if you are ready to start, I will disappear for a while.
Okay, all right. Well, thank you, Alina, for the introduction. And you're probably the first person that's ever put amazing in front of my name, so I appreciate that. Um, the topic for the presentation today is about dry floor sanitizers. And as you can see in the title, I've emphasized the word science because this is meant to be a technical presentation. It's not a commercial marketing presentation. I'm not trying to promote a product or products. It's really about trying to share some of the learnings that we have with regards to these interesting products. Now, I've been working with biocides for over 25 years, and until I joined Sterilex, I had no idea these products even existed. And so for me, it's been a lot of fun working with them and learning about them, particularly on the efficacy side, because that's the thing that I'm most interested in. And so we're going to share some of that information as well as chemistry and other things about these products. So if you like technical presentations, I think you'll like this one. If you don't, I'll try to keep it interesting anyway. So with regards to an outline, I'll first talk about the importance of microbial contamination on floors. Then I'll talk about what dry floor sanitizers are and why they're used. Then I'll go into the application, where and how they're applied. And then I'll get into chemistry and formulation, which is not exactly my strong suit, but we do have our chemist on this call as well. So if you have any questions, she'll be able to answer those. And then I'll get into the stuff that I really am interested in, and that's efficacy. And that is whether we're talking about lab-based methods or field trials. All right, so why should we care about floors? I think most people on this call probably understand this, but studies have shown that floors are a high risk area for pathogen contamination, even dry floors. And it's not too hard to understand why. You have foot and vehicle traffic bringing things in. You have soils that are brought in with that foot and vehicle traffic, or soils that get deposited on the floor from various parts of food production. You also have irregular surfaces with cracks and crevices, which make nice little habitats for microorganisms to hide out in. And then last, but certainly not least, you have drains, which I think, again, probably most people on this call understand are a very good reservoir for microbial contaminants. In this little figure that I have, this came out in a publication in 2023 from the European Food Safety Authority. And what it was looking at was persistence of a variety of pathogens. In this case, it's Listeria And they were looking at all the studies out there about persistence, where is it occurring? And they looked across various industries, dairy, meat, fish, and seafood, as well as various locations. And you can see that I've circled two areas. One is drain and one is floor. And the size of those bubbles that you're seeing, that indicates the number of studies indicating persistence in those areas. So you can see, for example, in meat production, that drains and floors pop out a lot for Listeria persistence, as does some other areas. For example, in fruit and vegetables, drains pop out. So again, this is just kind of to emphasize the importance that floors and drains can serve as a reservoir for pathogens like Listeria. Now, you may be asking, well, that's my floor. I don't do food production on the floor. Why should I be concerned about what's on my floor? And I think you also know the answer to this too, and that is because what is on the floor may not stay on the floor. And I give a couple of examples of this. In the picture on the left, this was from a study back in 2006, and they were looking at oil meal production and salmonella contamination. And in the picture, what you're seeing is what's called a pulse field gel electrophoresis. And what that was usually, well, has been typically the way that people would type salmonella strains. And what they're showing in this picture is that the same salmonella pulse field type is showing up in multiple locations. So it's on the floor, it's on some boots, they found it on some rodents, and then they found it in the product itself. And their conclusion from this study was that within an oil meal plant, high salmonella contamination rates for the floors represented the greatest risk of contamination for the oil meal. And that was occurring via operators, dust, air, and even rodents. And so they really emphasized that controlling contamination on the floor was critical for controlling salmonella in the oil meal product. Another example of how things can go from floor to other areas is shown in the table on the right. This was a study done by the USDA Agricultural Research Service. They published this in the Journal of Food Protection back in 2012. And what they did, they set up a neat little experiment where they created a drain environment where they made a biofilm of Listeria on the drain, which was what they call their empty pipe spray. And then they also made basically a Listeria culture in the drain itself, in the liquid. And then what they did is they sprayed it for 2 seconds with a hose, and they put various auger plates around this drain, and they wanted to see how far out Listeria would go after they sprayed the drain. And what you can see is that if you just look at the floor and the wet pipe spray, they were able to detect Listeria 4 meters away from the drain. And then not just horizontally, but vertically, they could see Listeria spreading. And so when you see the bench, the site identified as a bench, that's an elevated site. And as far out as 2.5 meters, they could see Listeria at an elevated level away from the drain. Again, simply by hitting that drain for 2 seconds with a spray. So I think you can understand that just because it's on the floor doesn't mean it's going to stay on the floor. Now, there are a number of wet chemistries out there for treating floors. What I'm going to talk about today is a dry type product, a dry floor sanitizer. And what this is it's a dry powder or granule that applied to floors that when it's activated by moisture, and we'll talk a lot about this moisture part, exhibits antimicrobial activity in the surrounding environment. The actives and inerts vary in these products, and we'll talk some more about that when we get into the chemistry. And they're used primarily for two reasons. One is to sanitize and disinfect the location where they're applied, for example, around a floor drain. You can see that in the picture on the left. But another way that they're used commonly is to reduce the spread of microbial contamination from one location to another via foot or vehicle traffic. So think more like a foot pan or a tray, or as you see here in the picture, an area where foot traffic is going to occur or even forklift traffic. So common application areas for these products include foot pans or mats, high traffic areas, forklift routes, doorways, loading bays, and again, last but certainly not least, drains. Applying these products does not require any kind of sophisticated equipment. You can see in the picture in the upper left, somebody is applying powder with a simple scoop, and they're putting it at the door of a refrigeration unit where a lot of condensation tends to land on the floor. If you look at the picture in the upper right, here they're using a handheld spreader, similar to what you might use to spread fertilizer in your yard. And then if you had a larger application, you might use a larger fertilizer type spreader, which is shown in the lower left. And then you can see also some people will put it in a pan or a tray, which people will walk through as they go from one area to another. Now we get more into the science of these products, and I'll start with chemistry and formulation. So these products have an active ingredient, and then they have co-formulants, and we'll talk about both. With regards to actives, the most common that you'll see out there are oxidants, and among the oxidants, the most common is hydrogen peroxide. But we're not talking about hydrogen peroxide as a liquid. We're talking about it typically in the form of sodium percarbonate. And if you look at the little equation, chemical equation on the right, you can see that sodium percarbonate has hydrogen peroxide associated with it, and then it'll dissociate and form hydrogen peroxide or release hydrogen peroxide, which is a good sanitizer disinfectant. However, hydrogen peroxide does have what I would call a little bit of an Achilles heel, and that is there are a lot of bacteria out there that make an enzyme called catalase, and they can break hydrogen peroxide down to water and oxygen. And so if you have a low level of hydrogen peroxide and you have a very active catalase population, you could see that peroxide dissipate fairly quickly. Another oxidant that you'll see commonly used is peracetic acid. Again, we're not talking about liquid peracetic acid. In this case, the peracetic acid is going to get generated in situ. And typically what the product will have, it'll have a source of hydrogen peroxide, like sodium percarbonate, and then it'll have another molecule. In this case, it's TAAD. And when the two mix, they will form peracetic acid in the presence of moisture. And so again, we're not talking about liquid PA, we're talking about PA that's going to get generated from two components in the powder when the powder comes in contact with moisture. And then we also see some chlorine-based products out there, not as common as the sodium percarbonate or PA products, but there are some chlorine-based products out there as well. Another active that you may see are the quaternary amines. I show a structure of that in the lower right. So you will see some of those in products as well, but usually more oxidants than quats typically in these products. Now, in terms of the modes of action, oxidants oxidize and inactivate various cellular components. They're very broad acting. And many people don't know this, but bacterial cells and actually eukaryotic cells as well are anaerobic inside. They're reducing. And so when an oxidant comes in and starts to destabilize that anaerobic environment, it really wreaks havoc on the physiology of a cell. So that's why I say they can be very broad acting. And that's also why bacteria and our own cells have so many defense mechanisms for oxidation. Quaternary amines tend to be a little more specific. They attach to and destabilize bacterial and other cell membranes, even though there's been some recent data to show that they have other functions as well. But typically, their primary activity is in destabilizing cell membranes. Now, coformulants. So these products, again, a typical product is not just going to have an active ingredient. It's going to have other components. And what I have listed here are some of those other components that you might see. So there might be a pH adjuster to maintain a proper pH. There might be wetting agents, and these are designed to reduce surface tension of water. So when the powder gets wet, it'll allow for the actives to spread out over the surface and enhance contact with microbes. But these wetting agents can also impact antimicrobial activity, and I'll talk about that on the next slide. Typically, you might see a chelant in these products. They're designed primarily to increase the stability of the active, especially if in the presence of hard water. But like the wetting agents, they may also affect antimicrobial activity. Chelants are very good at destabilizing the outer membranes of gram-negative bacteria. They're also very good at destabilizing biofilms too. There might be a processing aid, a deduster to reduce dustiness. There might be an anti-caking or flow agent to prevent caking of the product, especially if it's being stored in a really humid environment. Other coformulants can include colorants for safety so that the product will be distinguishable from, say, a white food ingredient. And then also these coformulants may help in terms of traction enhancement. Now, I mentioned that things like wetting agents may also, and chelants may also have an effect on antimicrobial activity. And that's what I'm showing in this graph. So regulatory agencies in both the US and Europe recognize that coformulants may affect the activity of other or of active ingredients. So it's well recognized. And in the BPR, you will see actually guidelines for coformulant testing, which basically allows for some level of activity from the coformulants, but not too much. They don't want the coformulants to be the actives. So what you're seeing in this graph is an EN1276 test. I'll talk a little bit more about the types of tests in a minute. This is a suspension test where we're testing a dry sanitizer against Staph aureus. And what you can see on the bottom is the different types of formulations that were made. And then on the y-axis is the log reduction. And then the different colored bars represent different contact times. So if we have a product that only has sodium percarbonate in it, okay, so this is just the active, it's making hydrogen peroxide, you can see that given enough contact time, we can achieve the desired level of kill, which in this case is 4 or 5 logs, sorry. But it required 60 minutes. If we start to co-formulate this product with wetting agents, surfactants, and chelants, we can now start to move that contact time down. And you can see in Formula 3 on the far right, we've gotten the contact time down to 10 minutes, whereas if sodium bicarbonate was used alone, it was 60 minutes. So same level of kill, but now we can get a little bit quicker by incorporating things like surfactants and chelants. And so that again is one of the potential benefits of having coformulants in these products. Okay, so we'll get to my favorite topic, which is efficacy, which as a microbiologist, these are the things that we're most interested in. And I'm going to start on first a little bit about the regulatory aspect of these products and liquid products as well. So in the US, these types of products are regulated by the United States Environmental Protection Agency. So if we want to sell a product in the US, we have to conduct certain tests depending on what claims we want to make, and then the EPA either accepts it or doesn't. In the EU, we're dealing with the BPR. For BPR, this type of product would fall under the category of PT2, which is basically hard surface disinfection, and possibly PT4 if it's being used near food production. So for these categories, these product test types, there are specific tests that you have to conduct, and these are EN standard test methods. And really the most common are EN 1276 and 13697. 1276 is a suspension test, 13697 is a carrier test. And there are 4 bacteria that you have to test against, and you'll see those in a minute. Then you have to test in the presence of a soil. You have to work within a certain temperature range, and then you have a pass criteria. So in the case of 1276, you have to have at least a 5 log reduction to pass that test. And then there are other tests. For example, there's tests for candida, yeast, but usually these tests are really just modifications of the other 2 tests. So really this suspension test or the carrier test are kind of the critical tests for BPR. So here's an example of what some data might look like. So here, this product is an in situ PAA product that's being tested against the 4 standard organisms, 2 gram negatives, Pseudomonas and E. coli, and 2 gram positives, Enterococcus and Staph. And these are both tests, the suspension test and the carrier test. And what you can see is we looked at different rates of use of ranging from 25 to 75 grams per liter. So the way the product is tested is it's dissolved in hard water, and then it's tested like a liquid in this type of application. And what you'll see is that in this case that we failed at 25 grams per liter, but we passed at 50 grams per liter. And this was done under dirty conditions, which is a certain level of BSA with a 5-minute contact time at room temperature. So since we failed at 25 grams per liter, we have to make our claim at 50 grams per liter. So for example, for this product, on our label, it would say use at a level of 50 grams per liter. So if you're thinking about a floor and a dry product, how does that actually get translated to use? And I had to think about this a little bit. What does 1 liter of water on a floor look like? Well, if you look at the picture in the upper right, that's what 1 liter of water on a floor looks like. And I was given this presentation. I used this joke last time and nobody laughed. And I suspect nobody's laughing now, but I'm going to keep using this joke until I do get at least one person laughing at it. But in reality, that's not what we're doing, right? We're not applying the product to a beaker of water on the floor. Well, it might interest you to know that if you think about a square meter area and water to a depth of 1 millimeter, which is not a lot of water, that actually is a liter of water. And so for that, you can kind of construct, okay, if I have water that I think is approximately this depth and I'm treating this amount of area, then you can understand how that 50 grams per liter can then easily get translated into how much product I need to apply to that area. And that's what you're seeing in this little table is that if you estimate that you have this much moisture on the floor and you're treating this much area, this is how much product you would need or how many kgs you would need to apply to the floor. So these standard test methods do mimic certain types of applications of powder, and that is really applying it to a wet floor. Which begs the question, what about if I'm not applying it to a wet floor? What if I'm putting it on a dry floor, whether it's in a tray or I'm laying it down in a broadcast application, and my interest is reducing cross-contamination? Do these test methods mimic that type of application? And the simple answer is no, they don't. So one of the things that my group has been working on at Sterilex is to develop practical test methods that really mimic how these products are being used in actual production environments. And one of those methods that we developed, we call the mini boot method. And what this method is designed to do is measure the effectiveness of a dry floor sanitizer in reducing cross-contamination. And the way it works is we start with these little boot stamps, which you can see in the picture in the upper right. And what we do is we'll inoculate those boot stamps with a pipetter. And that inoculum will have a bacterial culture and some soil, because it's in any test method that you're working with, there's always going to be some kind of soil added to that test to mimic dirty conditions, if you will. We then take those boot stamps and we walk them through a Petri dish containing powder. And then we walk that stamp through 3 additional Petri dishes, which are empty. And you can see that in the upper right. You can see the first and the lower Petri dish. You can see the powder where the boot went in. And then you can see the Petri dishes that have been stamped with the boot after it goes out of the powder. And we have a control. The control is simply no powder. So we would walk it through an empty Petri dish and then into 3 subsequent Petri dishes. After 5 or 10 minutes, we then add a neutralizer to these Petri dishes, not the one that has powder in it, but in the 3 subsequent Petri dishes, because what we're looking for is the reduction of contamination after something's gone through the powder. And then we'll neutralize, we'll recover the surviving bacteria, we'll do serial dilution and plating to determine the bacterial count. So here's an example of some data. This is with a quaternary amine sodium percarbonate powder. So what you're looking at is data for Listeria and Salmonella. In the control, which again is no powder, you can see that in the first Petri dish that's walked through, and the second Petri dish, which we're calling the second step, and the third Petri dish, which we call the third step. So when you look at the control, you can see there's a high level of Listeria there, as you might expect, because there's no treatment. But if I walk it through the powder first, you can see that we've dropped down the Listeria count to our detection limit. So really close 6 to 7 log reduction. And with Salmonella, about a 6 log reduction, but the same sort of thing. Now, does every dry sanitizer work this well? And the answer is no. We've evaluated several dry sanitizers with this test system, and I would say that at the very least, we see a 2-3 log reduction. And then with some products, we see this level of reduction. So again, with any product we've evaluated so far, we at least see a 2 or 3 log reduction, but not always this level that we're seeing with this particular product. Okay, so another question we get asked all the time is, what if there is no water? And my first response to this is, are you sure there's no water? So if you put a dry floor sanitizer on a floor, a dry floor, and somebody walks through it with a wet boot or some other instrument or you know, a forklift that's got some water on the wheels, you've added water. So it's not a completely dry environment. But let's say it is. Let's say no water is getting introduced in any way, shape, or form. It's going on a dry floor, and the only potential for moisture is, say, relative humidity. Can the powders do anything under those conditions? And the short answer is yes, but it takes some time. So what we did here is we constructed another method where we prepare a bacterial inoculum with, again, a soil load. In this case, it was Cronobacter and milk. We dry it on a stainless steel carrier. And so you can see the inoculum application in the picture in the upper right. And then we spread powder over that carrier. Well, I'm sorry. First, we dry the inoculum so that there's no residual water there. And then we spread the powder over the inoculum. So it's a dried inoculum. And then we store those Petri dishes in various environmental chambers at room temperature and various levels of humidity. So the example I'm showing here, and we've done this with Listeria, we've done it with Salmonella, we've done it with a variety of organisms. I'm just showing you the data with Cronobacter. So on the y-axis, you're seeing the log reduction. And then on the x-axis, you're seeing the different levels of humidity and then the contact times. And so at 4 hours and 24 hours, we take the carriers out, we neutralize them with the powder on them, and we validated the neutralization, and then we do serial dilution and plating, and we look how many bacteria are present. So if there's no powder, and you can see the data in the blue bars, the log reductions, unlike the mini boot, you're going to see some level of kill, even if there's no powder, because as the organisms continue to dry out, so even though the inoculum looks dry physically, there's still probably some moisture there. And as that continues the drying process, you can see that we're getting anywhere from a half to 1.5 log reduction, depending on the humidity, even if there's no powder. If we do have powder, we're now seeing higher levels of kill. And so in the case at 35% relative humidity with this organism, we're seeing anywhere from a 1.5 log reduction over what occurred with the drying. And then under 70% relative humidity, we're seeing even more. In the case of 24 hours, we're seeing at least a 3 log reduction higher than what we would see if we just let the organisms continue to dry out. So yes, these products do show some performance benefit if added to a dry environment and kept in a dry environment. But what you're not going to see is a 4 or 5 log reduction in 5 to 10 minutes. To get that, you're going to need to have some moisture present. So, okay, so that's the lab. Now we'll talk a little bit about the field. So a question that's come up recently is how do you monitor for efficacy in the field? How do you monitor performance? And it is a little bit different with the dry sanitizers, and that's why I put this slide in here. So as far as sample location, swab, sponge type, testing method, that's usually going to be determined by the customer and their testing lab. So it's not like we come in and say, oh, you got to use this swab or you got to use this test method. That's determined by the customer. I mean, we might provide recommendations as far as where the products, you might want to apply them based on critical control points and all that sort of thing. But the testing is usually pretty much the same testing that the customer always does. The difference with the dry sanitizers is that with a liquid product, typically what's going to happen is you're going to apply it and then you're going to rinse it off, and then you would do your swabs or your sponges or whatever you use. With a dry sanitizer, it's usually meant to be there for some time. And so you don't, so if you're going to sample, how do you do that if the product's still there? So what we recommend is that you swab areas likely to be impacted by the presence of the sanitizer. And you'll see an example of that in a minute when I show you some field data. We do not recommend swabbing the powder per se, unless you have a really good neutralization system for your swabs and you're confident that you've neutralized any powder. But again, generally we say avoid swabbing the powder directly. You want to swab areas that you're trying to impact when you apply the powder. And this is something that we've actually taken inside my lab too to look at more closely. We're doing some internal studies now to look at various swab neutralization systems that are out there to see if anyone works any better than any other one. And if from that, we get some data to suggest that, hey, even if you pick up a little bit of powder with this swab system, it's okay, then we might start making some recommendations. But for now, we're not doing that. Again, we're generally just saying don't swab the powder directly. Swab the areas that you're trying to impact by applying the powder. Okay, so the first case study I'm going to talk about is in a protein processing facility. This protein processing facility was using a doorway foamer. They weren't happy with it. It was frequently breaking down. It was applying chemistry inconsistently and creating a wet and slippery environment. And so they converted to a dry sanitizer, and they were applying it to foot mats in the doorway entrances and directly to the floor for forklifts to drive through. And in the graphs, what you're seeing in the graph on the left, this is total plate counts. In the graph on the right, this is EB or Enterobacteriaceae counts. And here it gets to the point what I was trying to explain earlier is that they're sampling not where the product is, but away from where the product is. And in this case, it's 6 and 12 feet. And so with a boot foamer, you can see the counts that they got for TPC. With the dry floor sanitizers, the counts went down somewhat. And then for the EB is a little more dramatic in terms of the performance of the dry floor sanitizer versus the boot foamer. So again, relative ease of use, getting away from the problems of the door foamer and improved performance. And you can see why they were interested in converting to a dry floor sanitizer. Another case study I'll show you is one from a hydroponic grow house. This grow house was concerned with standing water from liquid sanitizers, and they were concerned that it was contributing to microbial growth. Again, like the previous example, they were concerned about cross-contamination from forklifts entering and exiting the grow house. So they started a dry sanitizer trial at the entrances into the plant using a hand spreader. And you can see this too is also looking at, well, not total plate counts, but aerobic bacterial counts, probably the same thing. And then EB counts. And with a doorway foamer, you can see how the product performed both immediately and 1 week later. Here, they're not looking at the floor, though. They're actually looking at what went through either the foamer or the dry floor sanitizer. So in the case of aerobic bacterial counts, they drop down to below detection with the dry floor sanitizer. And then in the case of EB, there really wasn't much there anyway, even with the foamer, but you can see one of them went down, but mostly there wasn't much there to show improvement with anyway. But again, they did see some good improvement with the aerobic bacterial counts. So this water thing, I've added this slide to kind of emphasize is one of the things that is driving people to use dry floor sanitizers, particularly for bath tray type applications, is their concern about liquid foot baths. there have been a number of publications, some of which I'm listing here, that have actually gone in and shown that if a liquid foot bath is not properly maintained, it can actually serve as a reservoir for contamination, not a reducing cross contamination. And granted, some of these studies are a little old, but in the first one, they actually found listeria in these foot baths. So not exactly what you want to do if you're trying to reduce cross contamination. So again, one of the advantages of the powders is you don't have to worry about something like that. The next and last case study is one from a protein processing facility. Here, their issue is that they were getting a high percentage of listeria positive swabs. So if you look at the graph, what I'm showing here is in the blue line is the total number of samples that they took at various times before the trial, which started back in the third week of October. And so you can see how often they're taking samples. And then the purple line is showing how many of those samples are coming up positive for listeria. And in this case, if you do the numbers, it ends up being a little over 40% of all their total swabs were coming up with presumptive listeria positives. We started the dry floor sanitizer. In this case, it was broadcast application to various locations. And you can see they're still taking a lot of samples, but the number of those coming back as listeria positives have dropped dramatically and is averaging at about 4%. So as you look at this time period, and that's the only time period we have data for now, we went from around 40% listeria positives to about 4% listeria positives using the dry sanitizer. Okay, so to summarize, dry floor sanitizers or dry powders or granules applied to floors that, when activated, exhibit antimicrobial activity in their surrounding environment. They sanitize and disinfect the location where they're applied. So if you're thinking about a drain, that would be an example. But again, they're also used to reduce the spread of microbial contamination from one location to another via foot or vehicle traffic. Location for use, foot pans, entryways, drains. Again, these products are formulated products, just like the liquid products that you may be used to. So you're going to have an active ingredient and coformulants, and those coformulants will have various functionalities. Efficacy, standard test methods are used, again, for registration and labeling, but may not accurately reflect the use of these products. And we're seeing regulatory agencies, both in the US and the EU, saying, hey, we really want to see some methods that more accurately mimic how these products are used. And so this is a thing that we continue to work on in my lab, is about how to develop methods to mimic how the products are being used. So you'll hear more about this if you follow this type of product. We realize that field results ultimately determine success. As a longtime microbiologist working with biocides, I've been involved in both. I'm partial to lab studies because they can be very controlled and I can clearly show benefits or lack thereof. Field trials, as probably most of you know, can be a little complex. Other things happen and so on and so forth. But ultimately, we realize that you have to see performance improvement in the field, or there's no reason to purchase the product. Okay, I just want to do some acknowledgments. I want to thank Alina and Nemus for letting me speak today. I want to acknowledge the Sterlex microbiology team, which is in the picture on the right. They're the ones generating all this data that I got to share today. I also want to acknowledge Robin Colas, who's on the call. She's our principal scientist for dry sanitizers, and she's our chemistry expert. So if you have any chemistry questions, I'm going to probably defer to her to answer those. I just want to also thank Sterlex Marketing for helping me put the slides together. And then my boss, Scott Burnett, longtime food microbiologist, who's really helped train me on food microbiology and antimicrobial applications in that environment. So with that, if you have any questions, I will try to answer them. If not, I may defer to some other folks that are on the call from Sterlex.
Thank you so much, Bruce. And I have to say that actually we will not let you go just like this because there are already quite a lot of questions.
Sure.
So, if you don't mind, I will start because basically we will have just 20 minutes left for our QA session. And I have also a question which I would love to ask. So, let's start with the first one. It comes from David. Could you use a warm water rinse of the same sample area, check pH with pH paper, and then swap? I'm sorry if I broke the pronunciation. I can repeat again.
No, I see what you're saying. So you're basically saying if I want to do an environmental swab to evaluate performance, could I basically wash off the powder and then swab, right?
Yeah, yeah,
Okay. Um, yes, I would have some concerns about the water source that you're using because obviously water can be contaminated. And if you're rinsing with contaminated water and it's there, then, my part of my questioning would be if you come find something, is what you found something that was still on the floor after powder application, or was it from the water itself? So that would be my biggest concern about rinsing off the powder, and then trying to do an environmental swab.
yeah. Thank you. And the next question is coming like, does the powder dissolve after activation?
Yes, it does. It'll dissolve as water is applied. So we will typically on our labels say that we will have a use rate, and if at some point the powder is gone, then obviously we recommend reapplying it. So depending on the level of moisture, level of water on the floor, you may consume the powder quite quickly or not. And so we do have in our labels, and we will make recommendations about reapplying depending on how much dissolution and use of the powder is occurring as after it's been applied.
Yeah, and is the powder working against biofilm?
That is something we do a lot of biofilm work in my lab. We've never looked at that. I would say that the potential is there. It would be it would be Yeah, I mean, it's something we should probably look at in the lab. I mean, the actives that are used in these products will have biofilm activity if they're used at a certain level. Can I say specifically that a powder applied to a floor with biofilm on it is going to remove or kill the biofilm? No, we've not done that type of testing. I would be interested in doing that type of testing. It just not has been real focus for us right now.
Yeah, thank you, Bruce.
Mhm.
Another question is this not about control of listeria in a food processing environment where it is very likely to exist in drains?
Mhm. Wait a minute, can you say that
Well,
again?
probably it was a question clarification.
Yeah, that's
So
what I was trying. Yeah, that's what I was trying to understand the
yeah,
question.
yeah. Is this not about control of listeria in a food processing environment where it is very likely to exist in drains?
Yeah, I mean,
So
it certainly is very likely to exist in drains, and these products are not meant to be the end all and be all in terms of sanitization and disinfection. They're designed to enhance performance of your overall sanitization and hygiene program. So we're not expecting somebody to apply dry floor sanitizer and take care of all their microbiome problems all over the plant. We're saying, look, these are some places, again, if some one of our people were to go into your plant, they would say, hey, these are some critical control points where you think you should apply powder, especially high traffic areas where you may be bringing in contamination. So again, nobody's saying that these products are the be all and end all in terms of sanitization hygiene. We see them as really improving upon those kinds of programs. And that's why drains are a big focus, because we know that drains are a problematic area.
yeah, another question is coming from Kevin. Does the powder present any sleep hazards? Hazards.
I'm going to let Robin answer that. She's more of an expert on the physical properties of the powder. Robin, can you talk a little bit
Uh,
about
yeah,
traction?
you can Robin, you can write it down, and I then. read
Okay.
it out loud. Yeah. So then the
So in
next
general,
question.
yeah. So in general, no, but Robin can give you more clarification on that.
Yeah, sure. So, like, if you don't mind, I will maybe drop the email
Hmm.
if there is any, or you can drop it in the chat. So,
Okay.
yeah. Then another question, or is like we have quite a huge queue waiting. Yeah, next question is coming. Are there any concerns regarding chemical contamination to products since the spreadability of listeria has been tested? Has the spreadability of the chemical been tested too?
Yeah. So this is a question that comes up. So most of the areas where these products are applied are going to be away from food production areas. You think about a warehouse or foot pan. Generally, we would say that we would want to see them. So let's say if you're thinking about a conveyor or something that's near the floor, we would want to see the product at least a half meter, if not more, away from that area. So this has not been an issue, but nevertheless, we want to be careful that it's not too close to food production. And Robin's banging on my door if you want to try to answer the traction.
Yeah, actually, you can join in if why not?
Yeah, yeah.
If there is a
So you're,
possibility
you're
of.
gonna get that. You're gonna get the microbiologist and the chemist. I suspect this probably won't be the only chemistry question we have.
That's the best, actually. Academy cannot imagine something better.
So it's about traction. They want to know about traction and slippage, potential for slippage.
Yeah. So,
You want to come over?
Show us. Hi,
uh,
Robin.
I, we have generally tested traction with two types of tests. One would be like a qualitative assessment with different evaluators walking across the powder or a non-applied surface with dry or wet powder. And for our US products, we've actually found that this generally increases traction or judgment of traction when applied. We also will evaluate via coefficient of friction using a friction robot that moves across the surface. And in those cases, the powders have generally measured below the threshold for US OSHA measurement of affecting or like basically affecting slippage. So generally the comments that we've gotten is that it tends to improve traction, especially in wet areas.
Thank you so much.
Hmm? Okay.
Next question.
Okay.
Actually, Robin, you can stay because maybe there is something else
Yes, yeah, I'll
coming,
just
so
point
why
to Andrew.
not? There are actually not so much time left, so you are super welcome to stay if you're not super busy right now. So the next question, do you have any case study data that shows period of time that this product keeps pathogens down to an acceptance not detected levels?
I can't think of any off the top of my head. Um, Again, it's so. Are you thinking in terms of? So I'm going to lay down the powder in this area. I mean, if you're continually applying the powder, I would expect to have the same level of efficacy over time. Now, if you're applying it once and then removing it, and a week or 2 weeks go by, then no, I wouldn't expect to have residual activity and performance. So as long as you're keeping the powder in place at the proper levels, we would expect to have the same level of control as you would see initially in the case studies.
Mhm. Thank you so much.
Mhm.
And are there any limitations on the surfaces where it cannot be applied or it can work poorly, for example?
Do you want to talk about floor materials?
Yeah, we have compatibility charts for our products on our website, but there is some incompatibility with like cast irons, brass, Some these would not typically be like flooring surfaces, so most most types of flooring surfaces that you would encounter are generally compatible with our products. It's where we see incompatibilities is with sometimes like the footings of certain pieces of equipment that might be exposed to the product, or like other types of uh, uh, polymer materials. But we usually test compatibility with the most widely used flooring surfaces, and we'll publish that information with our products.
Thank you so much. Next question. Are there issues with the staff hands when they are taking off footwear as they leave production, touching their footwear, then leaving the area?
So they're concerned about what, taking the product into other areas because it's on their footwear, or
I think
Yeah,
you tend to
basically
get that gloves
with
when
their
you're taking off
hands.
your shoe.
Yeah.
yeah.
I mean, the general safety recommendation is that you wash your hands if you're in contact with the product. Um, I don't believe we've gotten any complaints
No,
about.
not that I'm
Yeah,
aware of.
yeah.
Great. And in terms of implementation of the sanitation, for example, are there any special case, for example, if you implement it on a huge facility, like from scratch, replacing the wet sanitation? What are the concerns to consider and what are the challenges?
I could speak to at least one concern. I mean, you do want to start with, I mean, you certainly would not want to lay this down over another liquid sanitizer because depending on what that is, there could be some serious incompatibility. So if somebody was converting from a wet program to a dry program, you want to make sure that the that whatever they were using in the liquid sanitizer is gone before they apply the dry product. So good rinsing, that sort of thing. Um, that would be my first thought. Anything that you
It's
could,
more probably related to management, like factory management, and probably the structure as well of factory. Like,
again, I think, again, it depends on what you're trying to do. And I think that's why it's important to have somebody come in. I mean, we have field people that come in and really work with, well, what's the problem? What's the issue? And what are you trying to do? And most plants are going to have some good ideas about where their critical control points are. So and that's where the decisions get made as to whether, do we need to do a broadcast application, do we need to have a foot pan, do we need to focus on this area or that area. So it's a little hard to answer a broad question like that other than with some generalities. Like I say, my first thought was, when you're talking about a liquid sanitizer and then going to dry, is we just would want to make sure that that liquid sanitizer is not there when the dry stuff starts to go down, that it's rinsed off. But yeah, as far as specifics about where and how, it really, it's always going to be plant dependent. And that's why it's important to have people come in and assess where the best use of the product is.
thank you so much, Bruce. Thank you so much, Robin, for joining in. And yes, I think it was the best kickoff ever of the 2025 academy season. And I just ask everyone who is actively listening us just to shoot on the reaction on the bottom, like shoot on some hearts, claps, whatever you want, because I'm pretty sure that it was really super useful presentation and you took a lot of out of it. And me, I'm just warmly inviting you for the next session, which will be already next week with a friend Yanis, where we will talk about food safety culture. Don't miss it out. Register if you haven't done it yet. And wishing everyone who had already finished his her working tasks amazing evening. And those who are still in the morning routine, have a lovely day and see you in our next Academy sessions. Bye.
All right. Bye bye. Thank you.
Thank you.